In type 1 diabetes, there is autoimmune destruction of the beta cells of the islets of Langerhans. These beta cells are responsible for producing the hormone insulin. Therefore, when these cells are destroyed, insulin cannot be secreted in response to rising levels of blood glucose. Insulin has many different physiological functions, but one of the first sites of action of insulin is the alpha cells of the islets of Langerhans that are responsible for producing the hormone glucagon. And high levels of insulin inhibit the secretion of glucagon from these alpha cells. Therefore, when insulin levels are low, the alpha cells are released from their inhibition and begin to secrete high levels of glucagon. This is important because glucagon opposes many of the functions of insulin. Therefore, this high level of glucagon complements the low levels of insulin in giving rise to the pathophysiology of diabetes. Now, another concept. As we shall go on to see, untreated type 1 diabetes exerts a significant biochemical and cellular stress on the body. This stimulates the release of certain hormones which are part of the stress response. These hormones include hormones like adrenaline, which is mainly synthesized and released by the adrenal medulla, noradrenaline, which is mainly released by the ends of the nerves of the sympathetic nervous system, as well as others, such as cortisol and growth hormone. As a group, these hormones are called the counter-regulatory hormones. Counter because they generally oppose the functions of insulin, and regulatory because high concentrations of these hormones inhibit the release of insulin. Because the amount of insulin that is produced by the dying beta cells is already low, these counter-regulatory hormones exacerbate the problem by decreasing the levels of insulin even more. So the concept I would like you to appreciate is that, yes, in diabetes there is low levels of insulin, but the pathophysiology is caused not only by the low levels of insulin, but the high levels of glucagon and the high levels of these counter-regulatory hormones. So from now on, whenever I say a decreased level of insulin leads to something, what I actually mean is that the decreased levels of insulin, increased levels of glucagon, and the increased levels of counter-regulatory hormones lead to that thing. Let's now move on to talk about some of the specific effects of decreased levels of insulin on certain tissues. The first type of tissue I'd like to discuss is striated muscle, particularly skeletal muscle. In normal physiology, when the levels of blood glucose are high following a meal, for example, the increased levels of insulin promote the expression of glucose transporters called the GLUT4 channel on the membranes of skeletal muscle cells. So when the levels of insulin are low in diabetes, this leads to decreased expression of the GLUT4 glucose channel on skeletal muscles. Usually, when the levels of blood glucose are high, these GLUT4 channels allow glucose from the blood to diffuse into the skeletal muscle, thereby helping bring the concentration of blood glucose back into the normal range. However, because a lack of insulin decreases the expression of these channels, skeletal muscles are less able to contribute to decreasing blood glucose concentrations back towards the norm. This plays a role in the hyperglycemia we see in diabetic patients. Now, I'd like to move on to talk about another concept. Insulin is actually an anabolic hormone. That is, when present, it stimulates the buildup of certain compounds in the body, for example, glycogen, protein and lipid stores. But when the levels of insulin are low and the levels of glucagon are high, this has a catabolic effect on the body. That is, it stimulates the breakdown of certain substances. In skeletal muscle, one of the things that are stimulated to be broken down is glycogen. And the breakdown of glycogen to glucose 6-phosphate is called glycogenolysis. Glycogen is the major storage form of glucose that exists in the body. Decreased levels of insulin promote the breakdown of glycogen into glucose 6-phosphate molecules in a process called glycogenolysis. This is important to appreciate because when a muscle undergoes intense or prolonged exercise, the amount of glucose diffusing in from the blood through these GLUT4 channels is no longer sufficient to meet the cell's respiratory needs. Under these circumstances, in normal physiology, the muscle breaks down glycogen to release glucose 6-phosphate that can be used to meet the cell's increased rate of glycolysis and respiration during exercise. However, in diabetes, these stores of glycogen are broken down. The muscle no longer has a reserve of glycogen to rely upon during prolonged exercise. This clinically presents as muscle fatigue. Now, decreased levels of insulin also stimulate proteolysis. And proteolysis is the breakdown of protein into amino acids. Proteins in skeletal muscle have many important physiological functions. Some of the most important proteins essential for muscle functions include proteins like the actin and myosin myofilaments, which, when they slide over each other, are responsible for generating the contractile force. Decreased levels of insulin stimulate increased breakdown of proteins such as these. As a consequence, these muscles are less able to generate the contractile force when they're stimulated. Clinically, this presents as muscle weakness. Muscle weakness and muscle fatigue are usually most prominent in the large, bulky proximal muscles, such as those around the hip or the shoulder girdle, because these are the largest muscles, they have the highest rate of metabolic activity, and are also used the most in day-to-day life. And muscle weakness and fatigue is often a general finding in patients with type 1 diabetes. Now, another concept. We said that the breakdown of protein releases amino acids, and these amino acids are released into the blood where they can be taken up by peripheral tissue such as the liver and used for gluconeogenesis, as we shall discuss in a moment. If you want a little bit more detail, the breakdown of protein in skeletal muscle yields a significant proportion of amino acids called the branched chain amino acids, or BCAAs for short. And the three branch chain amino acids are leucine, isoleucine, and valine. These amino acids are important because the skeletal muscle takes them and it removes the amino group from them and adds it to a molecule of pyruvate in a process called transamination. When an amino group is added to pyruvate, it generates the amino acid alanine, and this alanine is released into the blood where it's taken up by the liver and used as a substrate for gluconeogenesis, as we shall see later. In a similar fashion, The branch-chain amino acids are also used to synthesize the amino acid glutamine, which is also released into the blood. Particularly, it's taken up by tissues such as the kidney and the gut, and there it is used for gluconeogenesis as well. So to sum up what we've talked about so far, decreased levels of insulin decrease the expression of the GLUT4 glucose channel on the skeletal muscle membrane, and it also promotes increased glycogenolysis and increased proteolysis, which leads to muscle fatigue and weakness. The increased proteolysis, for a little bit more detail, also generates the amino acids, which can be used by peripheral tissue for gluconeogenesis. Now let's move on. The next issue I'd like to go on and discuss is the liver. The liver has many important physiological functions, but one of its functions is increasing synthesis and release of glucose when the blood glucose levels start to become too low. In diabetes, we know that the levels of blood glucose are in fact high, but the decreased levels of insulin force the liver into thinking the blood glucose levels are actually low. Therefore, the liver dutifully increases the synthesis and release of glucose from its intracellular stores in order to bring back the apparently low levels of blood glucose back towards the norm. And the liver has two mechanisms by which it attempts to do this. One mechanism is that the decreased levels of insulin increase the rate of gluconeogenesis in the liver. And gluconeogenesis is the synthesis of glucose from other compounds, such as amino acids and glycerol, for example. Amino acids are one potential substrate that can be used to make glucose in the liver, and there are many gluconeogenic amino acids that are capable of being used for this purpose. However, one of the most important substrates for hepatic gluconeogenesis is the amino acid alanine that is released from skeletal muscle. If you want a little bit more detail, when the liver uses the alanine released from skeletal muscle as a substrate for gluconeogenesis to make glucose, this is called the glucose-alanine cycle, and the rate at which this cycle happens also increases in untreated type 1 diabetes. Now, gluconeogenesis is a complex biochemical process that involves many different enzymes, and you won't need to know all of them. However, there are two enzymes which determine the rate at which gluconeogenesis happens. One of these enzymes is called phosphoenolpyruvate carboxykinase, or PEPCK for short. And the transcription of this enzyme from DNA seems to be one of the things that determine the overall rate of gluconeogenesis. Another important enzyme is called fructose 1,6-biphosphatase. The rate at which PEPCK is transcribed and the activity of fructose 1,6-biphosphatase determine the overall rate at which gluconeogenesis happens in the liver. Therefore, these enzymes are called the rate-limiting enzymes of gluconeogenesis. Decreased levels of insulin increases both the transcription of PEPCK and it also leads to phosphorylation and increased activity of fructose 1,6-biphosphatase, thereby increasing gluconeogenesis and the synthesis of glucose, which is released into the blood. Another mechanism by which decreased levels of insulin lead to increased glucose output from the liver is by increasing the rate of glycogenolysis in the liver. Earlier, we said that glycogenolysis is the breakdown of glycogen into glucose 6-phosphate. And again, this involves several different enzymes, and you won't need to know all of them. But the rate-limiting enzyme for glycogenolysis is an enzyme called glycogen phosphorylase. And this enzyme adds phosphate molecules to glucose residues on glycogen to break them down. This eventually leads to formation of glucose 6-phosphate at the end of the process. Decreased levels of insulin increase the activity of glycogen phosphorylase, thereby increasing the overall rate of glycogenolysis. Now, remember that we said the rate of glycogenolysis also increases in skeletal muscle. The glycogenolysis that happens in the liver is very similar to that which happens in the muscle. However, there is one important difference, and this is to do with what happens to the glucose 6-phosphate that is formed as a result. Glucose 6-phosphate cannot diffuse across the cell membrane through glucose transporters. Because of this, in skeletal muscle, all of the glucose 6-phosphate that is formed from the breakdown of glycogen is used for the muscle's own needs, and none of it is released by the muscle into the blood. However, in hepatocytes of the liver, there is an enzyme called glucose 6-phosphatase. And this enzyme removes the phosphate group from glucose 6-phosphate to make glucose. And this glucose can diffuse into the blood through the GLUT2 transporter on the hepatocyte cell membrane. And the reason why these hepatocytes possess the glucose 6-phosphatase enzyme is that, as we said, the liver has a responsibility to increase and maintain the levels of blood glucose when it starts to fall below the normal range, something which the skeletal muscle is not directly involved in doing. So to sum up, decreased levels of insulin increase the rate of gluconeogenesis and glycogenolysis in the liver, which increases the output of glucose from hepatocytes. This leads to even higher levels of blood glucose, and this acts to increase and sustain the high levels of blood glucose that we see in diabetes. The clinical name for a high concentration of blood glucose above the norm is called hyperglycemia. And I would like you to take away the concept that decreased insulin leads to exacerbation of hyperglycemia due to the effects it has on the liver. The next issue I'd like to go on to talk about is the white adipose tissue. In white adipose tissue, one of the effects of decreased levels of insulin is the decreased expression of the insulin-sensitive GLUT4 glucose transporter. This aspect is similar to the effect on skeletal muscle that we saw earlier. And decreased expression of the GLUT4 transporter on white adipose tissue means that glucose from the blood cannot diffuse into the adipocyte, and this therefore means that the adipocytes cannot help bring the blood glucose levels back down towards the norm. Another more important effect of the decreased levels of insulin in white adipose tissue is that it increases the rate of lipolysis. And lipolysis is the conversion of triglycerides that the adipocytes store into free fatty acids, and glycerol. Decreased levels of insulin increase the rate of lipolysis by leading to phosphorylation and activation of the enzyme hormone-sensitive lipase, which is the rate-limiting enzyme for this reaction. Because the stores of triglycerides is being broken down across all of the white adipose tissue in the body, clinically this presents as weight loss. Now, as we've been looking at, decreased levels of insulin have a catabolic effect. Not only do they stimulate the breakdown of stored triglyceride, they also stimulate the breakdown of glycogen and protein, for example. Decreased levels of insulin also prevent the efficient entry of glucose from the blood into cells through insulin-sensitive GLUT4 channels. All of these things mean that the amount of intracellular substrates in cells across the whole of the body are depleted. And it follows to reason that this gives rise to certain signals that lead to increased appetite. An increased appetite above the norm is called polyphagia. Which is increased appetite and increased intake of food, well above the norm for that person. Although it follows to reason that the weight loss and other catabolic effects lead to increased appetite, the exact mechanism by which this causes the increased appetite is still unclear. One of the theories is that when the amount of triglycerides in white adipose tissue is depleted, adipocytes decrease their synthesis of hormones such as leptin and adiponectin. Usually, high levels of these hormones act on certain areas of the hypothalamus, such as the arcuate and the paramedian nuclei, to suppress hunger. Also, one of the other theories is that certain areas of the hypothalamus and the limbic system possess insulin receptors or have insulin-sensitive glucose channels as well, whose activation helps suppress appetite in a normal healthy individual. Because the levels of these hormones is low, one theory is that this increases the appetite of a person above the norm, leading to polyphagia. Don't worry too much about this. The important thing to understand and appreciate is that increased appetite, but with paradoxical weight loss, is a classical symptom of diabetes and should always prompt suspicion of diabetes if these symptoms are elicited. I would now like to move on to talk about what happens to the free fatty acids and the glycerol that is released as a product of hydrolysis of triglycerides. The glycerol is a highly water-soluble molecule that's released into the blood and taken up by peripheral tissue, such as the liver, where it can be used as a substrate for gluconeogenesis, for example. Free fatty acids, on the other hand, are water-insoluble. So they travel in the blood bound to proteins such as albumin and are taken up by peripheral tissue, particularly by tissue which usually express insulin-sensitive GLUT4 channels and are unable to take up glucose in diabetes and therefore have to rely on free fatty acids as their respiratory substrates. One of the most important sites where the free fatty acids are taken up and used is the liver. And in the liver, these free fatty acids are metabolized and broken down to acetyl-coenzyme A in a process called beta oxidation, which occurs in the mitochondrial matrix. When the levels of insulin are low, not only does it stimulate the release of free fatty acids from white adipose tissue, it also stimulates their uptake and beta oxidation in the liver. One of the main ways this happens is that decreased levels of insulin indirectly increase the activity of the rate-limiting enzyme for beta oxidation, which is called carnitine acyl transferase 1, or CAT1 for short. And all this means the amount of acetyl-coenzyme A generated from beta oxidation of fatty acids is greatly increased. In diabetes, the levels of insulin are so low and the levels of glucagon and other counter-regulatory hormones are so high that the amount of acetyl-coenzyme A that is produced in the liver by beta oxidation is far more than the liver actually needs. So some of this acetyl-coenzyme A is used for the liver's own metabolic needs and, for example, enters the TCA cycle and aerobic respiration, but a significant proportion of it is excess. Now, these excess acetyl-coenzyme A that the liver does not need is used to synthesize molecules called ketone bodies. And these ketone bodies are released into the circulation where they can be taken up by peripheral tissue, such as the kidney, skeletal muscle, and particularly the brain. This is particularly important because normally the brain relies almost exclusively on glucose as its respiratory substrate. During periods of starvation, however, when the levels of blood glucose starts to decrease, the brain is unable to use fatty acids like many other peripheral tissues because fatty acids cannot easily cross the blood-brain barrier. So therefore, it relies on these ketone bodies that are produced by the liver as its respiratory substrate because these can cross the blood-brain barrier. So the reason why the liver produces ketone bodies from excess acetyl-coenzyme A is that, again, it has a responsibility to provide respiratory substrates to other tissues in the body, particularly the brain, when the levels of blood glucose are low, which it's fooled into thinking it is because of the low levels of insulin. Please don't worry too much about remembering this. This is just to give you some context behind why these ketone bodies are produced. The formation of ketone bodies from acetyl-coenzyme A is called ketogenesis. And when the levels of insulin are low, the rate of ketogenesis is increased. And again, ketogenesis involves several biochemical reactions involving several enzymes, but the rate-limiting enzyme is an enzyme called HMG coenzyme A synthase. And the activity of this enzyme is increased when the levels of insulin are low. Please do not confuse this enzyme with HMG coenzyme A reductase, which is a target for statins. This is a different enzyme from that one, although it has a similar name. Now, we said a little bit earlier about how carnitine acyl transferase 1, i.e. CAT1, was the rate-limiting enzyme for beta oxidation, and decreased levels of insulin indirectly increased its activity. If you want a little bit more detail, the mechanism by which this happens is that low levels of insulin decrease the activity of another enzyme called acetyl-coenzyme A carboxylase. And what this enzyme normally does is that it takes acetyl-coenzyme A and it adds carbon dioxide to it, i.e. carboxylates it to form Mellitus coenzyme A. And usually this Mellitus coenzyme A inhibits the activity of CAT1. But because there is a decreased levels of insulin, the activity of acetyl-coenzyme A carboxylase decreases, and this leads to decreased production of melonocoenzyme A. This means that less melonocoenzyme A is available to inhibit CAT1, and we can say that CAT1 is released from its inhibition. Depending on your syllabus, it might be worth knowing what exactly this CAT1 does. CAT1 is an enzyme that is present on the outer mitochondrial membrane. In order to understand what CAT1 does, it's important to understand that beta oxidation takes place in the mitochondrial matrix. Long-chain fatty acids, however, have a difficult time directly crossing the inner mitochondrial membrane to get to the mitochondrial matrix, because they are large molecules. What CAT1 does is that it takes a long-chain fatty acid, it removes the coenzyme A group from it, and it adds a molecule of carnitine. This complex of the fatty acid and the carnitine molecule is called acylcarnitine, and there is a specialized transporter on the inner mitochondrial membrane that is capable of shuttling this acylcarnitine into the mitochondrial matrix, where it can undergo beta oxidation. This special shuttle protein, if you really want to know, is called carnitine acylcarnitine translocase, and it's located, as we said, on the inner mitochondrial membrane. And CAT1, which is responsible for adding molecules of carnitine to fatty acids, is located, for your reference, on the outer mitochondrial membrane. But please don't worry too much about these details. I'd like you to just understand the concept that decreased levels of insulin indirectly increases the activity of CAT1, which is the rate-limiting enzyme for beta oxidation. And the excess acetocoenzyme A, which is formed as a result, is used to make ketone bodies, which also occur in the mitochondrial matrix. And these ketone bodies are eventually released into the blood. There are 2 or 3 ketone bodies which are produced, which you should know. One ketone body, which is produced as a result of ketogenesis in the liver, is called beta-hydroxybutyrate. Another ketone body, which is produced by the liver, is called acetoacetate. And if you really want a little bit more detail, this acetoacetate, when it's released into the blood, is a somewhat unstable molecule. So some of it chemically breaks down to form a ketone called acetone. And we'll see the relevance of acetone a little bit later on. Now, a few words of caution. If you know your organic chemistry, then technically speaking, you'll know that beta-hydroxybutyrate is not a ketone. However, in clinical medicine, it is classified as a ketone body, but don't worry too much about that. Also, this acetone is only a weakly acidic molecule that does not seem to contribute that much to the ongoing pathogenesis that these other ketone bodies cause, which we'll talk about a little bit later. So some textbooks omit the mention of acetone altogether. The concept that I would like you to take away now is that decreased levels of insulin increases the rate of ketosis and therefore lead to increased production of beta-hydroxybutyrate and acetoacetate, the ketone bodies. Usually there is some ketone bodies present in the blood. However, in untreated type 1 diabetes, the concentration of these ketone bodies rises well above the norm. An increased concentration of ketone bodies above the norm is called ketosis. And this can be detected either through specialized blood tests or through urine dipstick tests, because these ketones filter out into the Bowman's capsule and into the tubules. Because so much of it can be produced, not all of it may be reabsorbed, and some may appear in the urine. If you want a little bit more detail, usually the concentration of ketone bodies in the blood is less than 3 millimoles per litre. And ketosis, technically speaking, is when the concentration of ketone bodies is above this level. Also, if you want even more detail, Not only is the amount and concentration of ketone bodies increased in type 1 diabetes, also the ratio of beta-hydroxybutyrate to acetoacetate is changed. Usually, in normal physiology, the ratio of beta-hydroxybutyrate and acetoacetate is somewhere in the region of 1:1 to 3:1, so roughly speaking, they're equal. However, in type 1 diabetes, the ratio of beta-hydroxybutyrate to acetoacetate greatly increases. That is to say, the amount of beta-hydroxybutyrate that is produced greatly outnumbers acetoacetate that is produced. And the reason why we think this happens is because beta-hydroxybutyrate and acetoacetate can be interconverted, but the conversion of beta-hydroxybutyrate to acetoacetate requires the coenzyme NAD+. However, NAD+ is also required for beta oxidation. And because so much beta oxidation is happening, a significant proportion of this NAD+ gets converted into NADH. Therefore, there is less NAD+ available for converting beta-hydroxybutyrate into acetoacetate. And therefore, the levels of beta-hydroxybutyrate rises. And sometimes the ratio of beta-hydroxybutyrate to acetoacetate can be as high as 10:1, i.e. the concentration of beta-hydroxybutyrate can be 10 times higher than the concentration of acetoacetate in type 1 diabetes. But please don't worry too much about this. The reason why these ketone bodies are so important is that they can act as organic acids and therefore increase the concentration of hydrogen ions in the blood. One measure of the concentration of hydrogen ions is the pH scale. The higher the concentration of hydrogen ions, the lower the pH becomes. So when these ketone bodies lead to an increased concentration of hydrogen ions, this decreases the pH of the blood. Usually, the normal pH of the blood is somewhere between 7.35 and 7.45. However, because of the increased number of these ketone bodies, the blood can become so acidified that the pH actually drops below 7.35. And when the pH of the blood decreases to below 7.35, this is called acidosis. Now, these increased hydrogen ions also have another effect. Usually, the serum of the blood contains hydrogen carbonate ions, which are negatively charged. And one of the main purposes of these hydrogen carbonate ions is to act as a buffer to prevent sharp changes in the concentration of hydrogen ions. So as the concentration of hydrogen ions starts to rise, some of this hydrogen carbonate reacts with the hydrogen ions to form carbon dioxide and water. And what this process does, it prevents the increase of the concentration of these hydrogen ions and thereby allows the body to maintain homeostasis. However, in diabetes, there is so much ketone bodies that are produced, and this leads to a greatly increased production of hydrogen ions, which then actually start to deplete the amount of hydrogen carbonate ions in the serum. This leads to a decreased concentration of hydrogen carbonate in the blood. Usually, the concentration of hydrogen carbonate ions in the blood is somewhere in the range of 22 to 26 millimoles per litre. However, in diabetes, there is so much ketone bodies produced that this depletes the concentration of hydrogen carbonate ions to below this normal range. This combination of decreased pH below 7.35 and decreased concentration of hydrogen carbonate below the normal range is called metabolic acidosis. To give you some context behind what this means, if you're unfamiliar, there are many different things that can cause acidosis. But broadly speaking, acidosis can be divided into respiratory acidosis and metabolic acidosis. Respiratory acidosis typically occurs when there is insufficient ventilation of the lungs and therefore inefficient removal of carbon dioxide that allows the carbon dioxide to build up in the blood and react with water to form carbonic acid, which then causes the acidosis. Metabolic acidosis is generally caused by certain biochemical processes that lead to increased production of acidic compounds or ingestion of acidic compounds. The increased production of ketone bodies in type 1 diabetes is one cause of metabolic acidosis. Now, I'd like to move on to introduce another concept. This is the concept of the anion gap. The anion gap is simply the concentration of sodium ions in the blood plus the concentration of potassium ions in the blood minus the concentration of chloride ions in the blood minus the concentration of hydrogen carbonate ions in the blood. And the anion gap is simply a numerical sum of all of these. To understand why the concept of anion gap is important in our discussion, it's useful to appreciate that certain causes of metabolic acidosis give rise to a normal anion gap, but other causes of metabolic acidosis lead to an increased anion gap. The cause of normal anion gap metabolic acidosis are, generally speaking, gastrointestinal or renal pathology that lead to a loss of hydrogen carbonate ions, but also lead to a corresponding increase in the concentration of chloride ions. Therefore, there is little overall change in the concentrations of these anions put together, and therefore the anion gap remains roughly within the normal range. Causes of metabolic acidosis that give rise to a high anion gap are things like lactic acidosis, ingestion of certain substances, as well as the increased production of ketone bodies in type 1 diabetes. And these lead to a high anion gap because, as we discussed earlier, they lead to depletion of the concentration of hydrogen carbonate ions. Usually, the normal anion gap is dependent on the lab that is measuring it and the technique that is used. But a normal range is sometimes said to be between 6 to 12 milliequivalents per litre. However, due to the increased production of hydrogen ions by the ketone bodies in type 1 diabetes, this leads to such a depletion of these hydrogen carbonate ions that the anion gap increases above this normal range. This leads to increased anion gap metabolic acidosis, or simply called anion gap metabolic acidosis. So let's sum up what we've talked about so far. Type 1 diabetes increases the concentration of ketone bodies in the blood, leading to ketosis. These ketone bodies act as organic acid and increase the concentration of hydrogen ions and decrease the pH, leading to acidosis. Moreover, these hydrogen ions react with and deplete the concentrations of hydrogen carbonate ions in the blood, which allows us to call this acidosis metabolic acidosis, and which also leads to a high anion gap. This high anion gap metabolic acidosis that occurs secondary to increased ketone bodies that are produced as a result of type 1 diabetes mellitus is called diabetic ketoacidosis. And it is one of the most serious metabolic complications of untreated type 1 diabetes. And it should always be suspected in a patient who comes with high anion gap metabolic acidosis with ketosis, who is known to be diabetic or also has hyperglycemia. So that's it for this part of the lecture, and I hope you enjoyed watching. In the next part of the lecture, we'll be talking about some of the specific complications of hyperglycemia and diabetic ketoacidosis, and how they lead to other certain signs and symptoms.